So, and that's everything that he checks based on the quality scores. So this plot, and this plot are all based on quality scores. So you see that there's a lot more here that he checks. So for instance, this one. Apart from the quality scores, what is he also checking in your FASTQ file? Well, the second thing that he checks in your FASTQ files are duplicates. What are duplicates? These are reads with exactly the same sequence. And why is he searching for it? Well, because a lot of libraries for next-gen sequencing are contaminated. And this is a way to find the contamination. What is the philosophy behind it? Well, when you have a DNA-seq library, so genomic DNA, cut into pieces, what you typically do not expect there is that you will generate the same fragment multiple times. Because cutting genomic DNA into pieces is typically a random process. So you don't expect to see exactly the same fragment multiple times in these libraries. So, if you don't have exactly the same fragment multiple times in your library, you will also not have exactly the same reads in your FASTQ file. So if you see exactly the same reads in your FASTQ file, it typically points to these problems. You started from a small library and you had to do a lot of PCR to get enough copies to detect the signals. So a lot of PCR steps will generate PCR duplicates, duplicate reads in your FASTQ file. You can also have a cluster on the border of multiple tiles. It will be analyzed multiple times, once for each tile that it belongs to. So it will occur, it will generate the same sequence because it's one cluster, but it will occur multiple times in your FASTQ file. And then lastly, contaminations. I will come back on that. They will also give rise to a lot of duplicates in your FASTQ file. Why does he check it? Well, because for most applications, you don't want to see them. Yeah? If the duplicates are biological, of course you want to keep them. But if the duplicates are technical, then you don't want to keep them because they will distort your results. Because in every application that you do, at some point you have to count. And counting is influenced by these technical duplicates. So in RNA-seq, you do counting. Yeah, you know that. If you want to know the expression level, you have to count. But for RNA-seq, it's the only exception. Duplicates very often are biological. Because for RNA-seq, what are you going to sequence? You're going to sequence your messenger RNA. And your messenger RNA contains transcripts of housekeeping genes, like actin, and tubulin. Now, these genes are expressed in immense quantities. So you will have thousands and thousands and thousands of transcripts for actin in your messenger RNA. And if you're going to cut them, well, then it's no longer random. You have so many copies that even if you cut them randomly, you will generate the same fragment multiple times. So for RNA-seq, duplicates have a good chance of having a biological origin. You will also have technical duplicates, but you cannot distinguish them unless you use UMIs, but we're not talking about that. So for RNA-seq, they're often biological, so there it's the only case where you do not remove them. They can be biologically meaningful. But for variant analysis, where you have a genomic DNA library cut randomly into pieces, you do remove them. Because for variant calling, at some point you're going to count how many times a variant occurs. If it's just 1 or 2 times, it's a sequencing error and nobody believes that it's a real variant. If it occurs a higher number of times, then it's considered a variant, a real mutation. So you count, and these counts can be distorted by technical duplicates. And to avoid that, you remove them. And the same thing is true for ChIP-seq. In ChIP-seq, at some point, you're going to count. You're going to look for reads that occur more in your ChIP sample than in your control sample. That's counting. Eh? Also there, you don't want to include technical duplicates. And in ChIP-seq, you often have technical duplicates because the ChIP samples are very small. Yeah, the ChIP samples, what are they? They are the DNA that is bound by a transcription factor. Not that much of DNA is bound by a transcription factor. So you have small libraries, you have to do a lot of PCR there. Yeah? So in these two applications, you are going to remove your duplicates.